Hello everyone. In this lecture, we will try to get an overall idea about vaccine. So different types of vaccine, their advantage, disadvantage, how we can make them, etc. Okay. So first, what vaccine is? A vaccine is a biological preparation that improves immunity to a disease, and what a vaccine contains, it contains an agent that resembles a disease-causing microorganisms, and vaccine can be made from weakened or killed forms of the microbes. Okay, we can use the whole microbes, whether it's a killed or weakened, or we can use parts of it or its toxin. Okay, so the agent stimulates the body's immune system too. I mean, the agent that we use as a vaccine, it stimulates the body's immune system to recognize the agent as foreign and also destroy it and also remember it. Okay, so now a ideal vaccine, I mean, then a candidate that's we want to use as a vaccine, or you can say an ideal vaccine, should have some properties, some desirable properties. So what are those? An ideal vaccine should provide long-lasting immunity, should induce both humoral and cellular immunity, should not induce autoimmunity or hypersensitivity, should be inexpensive to produce, easy to store and administer. And also, vaccines must be perceived to be safe. Okay, we cannot use anything which is not safe for the target population. Okay. Yeah. And there are different types of vaccine, say live attenuated vaccines, inactivated vaccines, subunit vaccines, toxoid vaccines, conjugate vaccines, recombinant vector vaccines, DNA vaccines, RNA vaccines. Okay, so from the next slides, we'll try to get a little, I mean, more idea about these different types of vaccines. So first, killed vaccine. So in this case, we the normal wild type infectious pathogenic microorganisms are used as vaccine, but before using them as vaccine, those need to be killed or inactivated, and this can be done by treatment with using heat, formaldehyde, or gamma irradiation. Okay, so that they cannot replicate at all. I mean, in this approach, the wild type infectious microbes are killed, so definitely they will not be able to replicate at all. Okay, and thus will become non-pathogenic. Example of such killed vaccine is polio vaccine, typhoid vaccine, cholera vaccine, etc. Now, killed vaccine have some advantages. They are safe to use and can be given to immunodeficient and pregnant individuals, and cheaper than live attenuated vaccine, storage not as critical as live vaccine. And it also have some disadvantages, such as since the microorganisms cannot multiply, a large number are required to stimulate immunity. Okay, and periodic boosters must be given to maintain immunity. Okay, and only humoral immunity can be induced, and most kill vaccines have to be injected. Okay, cannot be given orally, rather they have to be injected. Okay, so then I'll talk about live attenuated vaccine, but before going to that, I would just like to talk about the how the B cell can get activated and produce antibody. I think you already know about this process. Okay, you have studied this in a different, in the basic immunology course. So just for a recap, I will try to explain it briefly. Okay, so how the B cell get activated, okay, as a part of humoral immune response. Okay, so please look at the photo, I mean figure here. So say this round one, it's the B cell. Okay, and we know the B cell, they have receptors. So with those receptors, they can bind with the antigen. Okay, say this B cells, it has a receptor for this antigen. So the antigen binds with the receptor, and then the receptor engulf this antigen, and digest it, okay, and then process it in their surface through the MHC molecules, major histocompatibility molecules. So once the antigen is presented by the B cell, then they attract the T cell, okay. So then the T cell come and they bind with it, okay. So after this, the T cell, they release cytokines, different cytokines, okay, and these cytokines, they can activate the B cells, and these activated B cells, then they differentiate and becomes plasma cells, and these plasma cells, they produce and release antibody specific to that antigen, okay. And this is the process how B cell, they can get activated by an antigen and then produce antibody specific to that antigen, okay. Okay, now get back to vaccine. So live attenuated vaccine, okay. This kind of vaccines is composed of live attenuated microorganisms that cause a limited infection in their hosts. So this limited infection is sufficient to induce an immune response but insufficient to cause disease. An example of such vaccines are measles vaccine, BCG vaccine, etc. And the advantages of live attenuated vaccines are infectious microbes can generate both memory and humoral immune response. I mean, if you can remember, the killed vaccine, they can only induce humoral immunity, but live attenuated vaccine, they can generate both memory and humoral immune responses. And as they are live, so this can multiply in the host, so fewer quantities must be injected to induce protection. I mean, we don't need to inject lots of vaccine to induce the immunity. So they are live. So if we can inject fewer quantity, so after this injection, it can multiply in the host body and can induce protection, that is immunity. So that's why multiple booster does may not be required most of the time, okay. And also the whole microbes stimulate response to antigens in their natural conformation, because we are using the live virus, okay. So I mean live microorganism, so that they can raise immune response to all protective antigens, okay. And some live vaccines can be given orally, which gives more protection at the normal site of entry, and oral preparations are less expensive than giving injections. Well, they also have some disadvantages, such as may very rarely, may very rarely revert to its virulent form and cause disease. I mean, though they are they are attenuated, but they may revert to its virulent form and thus can cause disease. And live vaccines cannot be given safely to immunosuppressed individuals, okay. And since they are live and because their activity depends on their viability, proper storage is critical, okay. So I mean, we have to keep the vaccine live, so storage is very critical, okay. Okay, so now then subunit vaccines. So subunit vaccines, they contain purified antigens instead of whole microorganisms, and in this kind of preparations, they consist only those antigens that elicit protective immunity, okay. Because I mean, a microorganism, they have both I mean, they have antigens. Some of those antigens, they are involved in causing the disease, some they are involved in induce the immunity, okay. So the I mean, the antigens that we will choose for subunit vaccines, they will be only those antigens that elicit or induce protective immunity, okay. And they can be composed of toxoids, subcellular fragments, or surface antigens, okay. And effectiveness of subunit vaccines is increased by giving them with adjuvants, okay. I mean, sometimes with the subunit vaccines, we need to add some adjuvants to increase their effectiveness or effectivity, okay. And I mean, how the adjuvants usually work: adjuvants slow antigen release for a more sustained immune stimulation, okay. And the advantages of subunit vaccines, they can safely be given to immunosuppressed people. They are less likely to induce side effects. And the disadvantages of subunit vaccines are antigens may not retain their native conformation, so that antibodies produced against the subunit may not recognize the same protein on the pathogen surface. Okay, so which means, sometimes the subunit vaccines, or the candidates of subunit vaccines, they induce the antibody, but those antibody may not work on the real microorganisms. Okay, and isolated protein does not stimulate the immune system as good as a whole organism vaccine. Okay, then conjugate vaccines. Okay, so primarily these kind of vaccines are developed against capsulated bacteria. Okay, and these are actually are subunit vaccine, and they stimulate only humoral immunity. That means a humoral immunity in a T independent way. Okay, so T independent humoral immunity. Okay, and usually they generate short-lived immunity. Actually, the conjugate vaccines, most of those vaccines are there. The vaccine candidate are polysaccharide. Okay, so uh, generally polysaccharides are less. Polysaccharides, they cannot induce very strong immunity. Okay, so they usually induce T independent humoral immunity. Okay, however, the effectiveness of conjugate polysaccharide vaccines can be increased by linking them with protein carriers. Okay, so when so we can actually this will act as adjuvant. We can add the polysaccharide vaccine with a protein. Okay, and then the T independent immunity induced by those kind of polysaccharide vaccines will be converted into T dependent antigens. Okay, and thus the protective immunity is induced. An example of such vaccine is Haemophilus influenza HA vaccine. Okay, so it's a polysaccharide vaccines. So, but to increase its effectiveness, this polysaccharide vaccines is complexed with diphtheria toxoid. That means the polysaccharide antigens, they are mixed with, or they are joined with diphtheria toxoid. So diphtheria toxoid is a protein. So thus the effectiveness of Haemophilus influenza HIV vaccine is increased. Okay. So as I said, the polysaccharide vaccines, they induce T independent response. Okay, so what T independent response means? So antibody production does not require assistance from T cells, and this kind of antigens are mainly polysaccharides or lipopolysaccharides with repeating subunits bacteria. Okay, and as the T independent response, usually they produce weaker immune response. Okay, so in comparison, T dependent response. So we can convert the T independent response given by polysaccharide vaccine. We can convert it into T dependent response by conjugating the polysaccharide with protein. Okay, so in case of T dependent response, antibody production requires assistance from T helper cells, and a macrophage cells ingest the antigen, and peptide antigen presents it to T helper cell. Okay, and the T helper cell then stimulates B cells specific for antigen to become plasma cells, and antigens are mainly proteins on viruses, bacteria, foreign molecules, etc. Okay, in this figure, okay, it shows how the this figure shows a immune response done by T helper cell. So for example, so this is a bacteria with T dependent antigens. Okay, so this bacteria, it can, say this is the bacteria. Okay, and this is the macrophage, and we know that macrophage is can act as a antigen presenting cell. So when this macrophage will engulf this bacteria, they will digest it and process it, and after the processing the macrophage will present the bacterial antigen fragments into their surface by class 2 MHC proteins. Okay, so once the antigens they are presented, the T helper cells get attracted and come and bind with the macrophages. Okay, so through CD4, and then this activated T helper cell they release cytokines such as IL-2 and other cytokines, and these cytokines activate the B cells, and the activated B cells they then can differentiate into memory B cells and also plasma cells, and the plasma cells they produce and secrete the antibody against the antigen. Okay, and then this photo, actually this photo shows the kind of these photos, the left side it shows how a polysaccharide vaccine induce uh, T independent they induce the immunity by a T independent way. Okay, and this right side it shows how we can add or conjugate a toxoid or a protein with these polysaccharide vaccines, and thus convert the T independent immunity into T dependent immunity. Okay, so let's see. So say suppose this is okay, meningitis A polysaccharide. Okay, this is an example of polysaccharide vaccine, meningitis A polysaccharide. Okay, so it can bind with the B cell. Actually, when it's bind with the B cells, it binds to multiple receptors at a time. Okay, and through this binding, they can induce the B cells, and the B cells they become differentiate into plasma cells and then produce antibody. Okay, so no memory cells are produced here. Okay, only the plasma cells are produced. So no memory cells are produced here. So that's why long-lasting immunity is not induced. Okay, is not generated in this case. But if we add a protein with this polysaccharide, so for example, tetanus toxoid. Tetanus toxoid is a protein. Okay, so in that case, so again, then this conjugated vaccine, it will or this conjugated antigen, it will bind with the B cell. Okay, the polysaccharide portion it will bind with the B cells through multiple receptors. Okay, and they will activate the B cell. However, the added conjugate, that's the protein. Okay, it will help this vaccine to to induce this conjugated protein. Okay, so it will actually help this vaccine or antigen to activate the B cells with the help of T helper cells, because in that case, when the only polysaccharide was there, okay, so this was not possible to occur. But once this protein was added with this polysaccharide, then this pathway was possible. So okay, so because of this toxoid, eventually the B cell they will they engulf this vaccine and digest them and process them in the surface by major histocompatibility complex. Then the T cell they will get attracted and bind with them, and then they will release cytokines which will activate the B cells, and these activated B cells then then can differentiate into both memory cells and plasma cells. Okay, now another type of vaccines, recombinant vaccines. Okay, so this kind of vaccines are produced using recombinant DNA technology or genetic engineering. So what is done? Genes for desired antigens of a microbe are inserted into a vector, that means a plasmid. For example, hepatitis B virus HBV vaccine is a recombinant subunit vaccine. In this case, the hepatitis B surface antigen is produced from a gene transfected into yeast. Okay, yeast cells and purified for injection. So what was done? First the gene which encode the hepatitis B surface antigen was selected, and then that gene was expressed, was transfected into yeast, and then the yeast cell, it will, as this its yeast cell now has the gene, so now they will actually produce the surface antigen, so we can just harvest and purify them and use them as vaccine. So in this photo, it shows the steps by which the hepatitis B surface antigen vaccine is produced using a yeast cell. So first we need to identify, or we need the gene that encode the hepatitis B surface antigen. Okay, and then we have to insert this gene into a vector or plasmid. Okay, so please remember, there are different plasmid which are used in, like, actually the plasmid that you will use will depend on the your or microorganism. Okay, so suppose you want to use E. coli as your expression system, so in that case you need to choose some different kind of plasmid. And then say you want to use mammalian cell, then there are some other plasmid. So now if we want to use yeast, in that case you have to choose those plasmids which express in the yeast, okay, abundantly. Okay, so all the plasmids, they don't express in all the organisms in the same way. Okay, some plasmid, they are optimized for the yeast, some they are optimized for the mammalian cell. So as in this case you're using the yeast cell, so the plasmid need to be optimized for the yeast. Anyway, those kind of plasmids are available commercially. So anyway, so first you have to you have to select the gene. You need the gene that encode your hepatitis B surface antigen, then insert it into the plasmid. Okay, so the plasmid is ready now. Then you have to transfect this plasmid into the yeast cell. Okay, and then you have to select means, after transfection or transformation, all the yeast cell will not have this plasmid. So there are screening method. Okay, so by which you can screen that whether there are screening method, and using those screening method you can specifically select the yeast which have the plasmid. Okay, so then after selection you got your yeast with the plasmid. So now then just culture them in large quantity, then harvest your cell. Okay, and lyse your cell and purify your hepatitis B surface antigen. Okay, because whenever you will culture this yeast cell, as it has the plasmid, it will produce the hepatitis B surface antigen. Okay, so this was the strategy or the steps by which hepatitis B surface antigen vaccine can be produced by yeast cell. Okay, by recombinant DNA technology. Okay, now the recombinant vaccines, they also have some advantages and disadvantages. First, advantages. So those vectors that are not only safe but also easy to grow and store can be chosen. Okay, this is advantage. Okay, so and then antigens which do not elicit protective immunity or elicit damaging response can be eliminated from the vaccine, because we are choosing the gene. So the antigens which are damaging, we just can omit those genes. Okay, then disadvantages. So since the genes for the desired antigens must be located, cloned and expressed efficiently in the new vector, the cost of production is high. And when engineered vaccinia virus is used to vaccinate, care must be taken to spare immunodeficient individuals. Okay, sometimes these recombinant vaccines, we use another virus as a carrier. Okay, so for example, vaccinia virus is a very famous virus. This virus has been frequently used to generate this kind of recombinant vaccines for other microbes. Okay, so when this approach are used, so we have to careful that although this vaccinia virus, okay, we are using this as carrier, that means it should be safe for healthy person, but the immunodeficient persons, they can be even at risk if we use the vaccinia. Okay, so we have to careful in those cases. Okay, so I mean, if you produce a engineered vaccine using vaccinia virus, you have to be careful with the immunodeficient individuals. Okay, okay, so that's all for this part. In the next part of this chapter, we'll learn about DNA vaccine and RNA vaccine. So thank you for your patience.